Good afternoon or good morning to those of you that are watching today. Thank you for the Mexico Urogynecology Conference for allowing me to have this opportunity to speak with you today on Allomend Duo. It's a natural alternative to the next generation of the treatment of pelvic organ prolapse. My name is Dr. Andrea Pezzella. I am a urogynecologist here in West Columbia, South Carolina. I am the medical director and CEO of Southern Urogynecology Wellness and Aesthetics as my practice for over 14 years. I've been a urogynecologist for 20 years, and I've actually seen the evolution, if you will, of the different options for pelvic floor reconstruction. And I think that I've been able to see and use all the different approaches to pelvic floor repairs, both vaginally and abdominally, with or without mesh or graft. And so I'm really excited today to discuss with you alternatives that I have designed and introduced in my own skill set, options for pelvic organ prolapse. These are my disclosures. So our learning objectives today are to review polypropylene mesh history, talk about Allosource as a company and their product Allomend Duo, which I'm currently using in my practice. And then I'd like to share with you my novel approach or my technique of the biologic graft in pelvic floor reconstructive surgery in my practice. So I'd like to go over and just kind of step back and talk about how grafts are being used. Here we could show with you today how the grafts have been considered in the anterior and posterior compartments. So these are two compartments that we have always considered very difficult to repair. There's been definitely discussion on what is the best repair and the evolution of how we go about approaching these two different compartments. We know the defect in the pelvic floor causes the bladder or rectum to prolapse through this weakened, torn, or attenuated pubovesical cervical fascia or rectus fascia, as in the posterior compartment. And so we're often faced with this durability issue, this issue with is it best to do a native tissue repair, or do we augment with a more durable material that can give strength and durability and actually allow us to achieve overall a reduction in our recurrence? So historically, we've attempted to choose a graft, particularly for the anterior compartment, because we've known that, and in studies show that there is a higher risk of recurrence in the anterior compartment, anywhere from 30 to 40%, depending on what literature you read. And so this area has been very controversial in terms of what to be considered. Pause. Okay.
Okay, go ahead.
So these is just examples of types of grafts that have been used. And historically, we've been using graft for over 50 years, starting off with hernia repairs. And then in the 70s, as early as the 1970s, we have used synthetic grafts. But there are different types of synthetic grafts as listed here. And then we also have a list of biologic grafts that we've used historically. Our synthetic graft most popular and what we use in pelvic organ prolapse recently is our Prolene macroporous monofilament type 1 synthetic graft. You have also Gore-Tex, Merceline, and these were very popular in the 80s and 90s to use. Our biologic grafts, whether we use porcine or bovine, and then autologous grafts, harvesting a rectus fascia graft or a fascia lata, and then the allografts, a cadaveric dermal or fascia lata. So mesh grafts have been used, and we also, in 2002, the FDA cleared the first surgical mesh products that became very popular for pelvic organ prolapse. Over the next few years, surgical mesh products for transvaginal prolapse were incorporated into what we call a kit. And these kits were where there was trocar-based utilization, where the graft was actually fashioned to fit the either anterior or posterior compartment. And you can see that there is all the tools and everything that you need, all the components that you needed for a complete pelvic organ prolapse repair. And these are examples where there was a very popular mesh graft kit. Particularly, I know in my hands, I used a lot of ProLift. I used Apogee Perigee initially, which then became Elevate. And so these were very popular and may or may not still be available in Mexico. So these different types of kits were used, and they were marketed as a very safe and reproducible option for pelvic floor reconstruction. Of course, there was a huge learning curve with these products, and unfortunately, it was placed in a lot of surgeons' hands that may not have had the skill set or were limited in training and had limited experience in pelvic floor reconstruction. And also, I don't think we really understood the biomechanics of the graft, which meaning how much tension to use on the graft. And that was really, I think, what was one of the downsides of, and the downfalls, I should say, of the use of mesh. So as we know, there was a huge mesh controversy, starting first with a warning in 2008, and then in 2011, where the FDA announced a statement regarding the increased risks of complications associated with mesh. So there was over continued concerns. There was over 3,000 complications reported in a short period of time, and the literature findings were very controversial. The FDA actually concluded that there were serious adverse events associated with the use of transvaginal mesh that were not rare. And the transvaginal mesh placement of pelvic organ prolapse actually did not conclusively improve clinical outcomes over a traditional non-mesh repair. Then in here in the US, there was increased litigation, class action lawsuits against the manufacturing companies mostly, but it also was a issue with the surgeons, and they started scrutinizing the surgeons. So the distributing manufacturing companies were losing millions of dollars, and the surgeons were getting lawsuits and litigation against them as well. So once this all happened, companies started to abandon the production of mesh, and some companies actually just dissolved. So they then by 2019, these transvaginal mesh kits were pulled off the market. So here in the US, it was a huge controversial issue, and everything halted. So since transvaginal mesh was no longer available, and I know it was, at least for me, a great tool, and I found in my hands and in my skill set, an invaluable material for me to allow for longevity, and its usability for me was great. However, I had to seek other options. So here I want to introduce to you um, Allomend Duo, which is an acellular dermal matrix. And I feel that bringing this into my hands, I believe that it was a nice alternative option for me, since I was not able to use transvaginal mesh anymore. Let's step back, though, and talk about Allosource as a company. Allosource is a vision-directed company. It is a nonprofit organization, and this procurement organization has a vision of leading the way in cellular and tissue therapies, and its mission is inspired by the gift of donation from our communities. And Allosource responsibly provides innovative cellular and tissue allografts to advance patient healing. And that's its mission. And so, as you can see, as a procurement organization, which has the first right of refusal, these are the stats, donor to patient. In 2023, Allosource actually had more than 8,300 donors processed and over 212,000 allografts distributed. And nationwide, over 70,000 donors processed and more than 3 million allografts distributed. So here are the facts, and this is just a testament to the reputation of Allosource. It is accredited by the American Association of Tissue Banks and registered with the U.S. Food and Drug Administration. It is one of the largest tissue bank donors here in the U.S. It has a 30-year history in biologics with 100 plus patents, both U.S. and international. It has over the country 37 ISO classified clean rooms, and it actually has over 200 types of allografts. There's 500 plus employees, and they process 365 days a year. And this is just their product innovation timeline, which just shows you how much Allosource is dedicated to not only musculoskeletal, but also dermal grafts. And this just shows you the evolution of how many different grafts that they actually produce, and where Allomend, its acellular dermal matrix, has been on the market for 15 years. So let me tell you, and let's talk about Allomend as a graft of Allosource and what differentiates it from other grafts that are currently on the market. It's the process. The process makes the difference. It has properties where it is unique, and it has a proprietary type of processing that is different from any other graft. It has a very strict criteria as far as harvesting the tissue. And again, it's procurement organization, which it has the first right of refusal. So they're very stringent as far as what donors they use. This has the ability to have a high quality of control, and it ensures its supply. And it's regulated by the Food and Drug Administration. But the process is what we call the DermaTrue decellularization process. It is a very gentle, dynamic cleansing process that allows all the cells to come out, your RNA and your DNA. However, it really preserves the tissue, and it prevents the damaging of the tissue. It also has a aseptic processing where the tissue stays fully hydrated, and the sterilization process where it has this what we call E-beam radiation, which allows the tissue a very high sterility assurance level. So this is all what makes the acellular dermal matrix different from any other graft. It also has a very precise tissue splitting. So the splits the thickness of the dermis into a very desirable exact thickening of the dermis. So it ensures consistency, and this just shows you the processing. And they also do what we call a 5-point of thickness verification, which is performed on every single graft. So this not only shows you a process where it keeps the graft very thick, but it also has where the thickness is throughout the whole graft. So you have consistency and durability. And this is just a high level overview of how invaluable it can be for us to use for our reconstructive procedures. So the graft can come mesh or non-meshed, and the meshed graft actually has and it allows for an increased surface area for potentially having a faster incorporation of your graft into the tissue. And also the meshed type of graft that they offer has a rapid fluid egress. So it helps guard against any seroma foundation. and it's flexible and pliable. So really provides optimal handling. So when you're using it in the operating room, these characteristics actually can be different from other grafts, and it allows you for easier use and then exact surgical placement. And it's ready to use. It's nice. It comes hydrated in the packaging and enables direct placement without having to rinse or have to soak the graft for any period of time. And it also has, of course, it minimizes the risk of infection through this sterilization process that I mentioned earlier. So you can have just quality and high standard of your graft. And then again, with this consistency of the thickness, it really helps me to ensure that I'm not going to have a thinner piece of tissue over when I fashion my graft in the arms, and it gives us a lot of durability, and it allows for me to have reproducible results. So as well as its consistency and the graft having, thickness throughout, it also has a dual-sided reticular dermal matrix. So you can use it, which we call non-directional implantation. So you can use either side. It's nearly identical, and you can see in this high magnification where you have structured on both sides of the graft, so you can use it on either side. So it doesn't have to have one side placed one way or the other. So it helps to increase your confidence while using this graft. And you can see the dual epidermal structure in 200 times and 1,000 times magnification. We can look at the Allomend as far as the scientific evidence. We have multiple bench studies where the scientific studies have shown that the surface area coverage and fluid egress properties are very viable. The biologic graft incorporates into the tissue and has high biocompatibility results. It also shows that the suture actually outbreaks the graft itself, so it's much stronger than the actual sutures. And it showed that there was actually sutures actually tore, or the anchor actually broke before the actual graft. And then its ultimate tensile strength was proven as far as holding and being very durable. So you can see that we have the scientific evidence that backs us up as far as utilizing this graft. So for people that need any kind of scientific evidence, we have that available. As far as Allomend specifically, as far as its clinical evidence, specifically used in pelvic organ prolapse and incontinence, Dr. Nathan Garrett has a retrospective study here, which shows short-term safety and efficacy with the use for transvaginal prolapse and incontinence repairs. And he had 54 patients that were followed over time, and really just showing its durability, demonstrating its use and safety. And also there were, in this limited study, 0 recurrence in this particular study. Now, what I'm going to show you today is what I've created as what my novel approach and technique to the use of the allograft. And I think many people have had their little nuances on how they use the graft, but I just wanted to demonstrate. And it's very similar to what I use and involved in using mesh, but I just transferred it over to the use of the graft. So this is so it's the description of my technique. This is a actual diagram of kind of a drawing of how I use my graft in my repair. You can see here that I have sort of the anterior wall, if you will, already opened up for us in that paravaginal space. And I have the full thickness vaginal wall here coming out, and I actually dissect that out into the paravaginal space. And then you have here is the bladder. But what I have, and I'll go over my steps, is this is the graft. I use a 6 by 12 centimeter graft, which is on my back table, and I actually um, fashion that very similar to what we've used in the past with like Elevate or ProLift into a graft where I have the arms that can reach back to the sacrospinous ligaments. And I'll refer back to this picture momentarily. So on my back table, and I fashion the graph, but before I fashion the graph, I want to make sure and ensure that I get into and I have my proper planes and I have my anchors in place. So before I open the graft, because it can be expensive, I do my hydrodissection. I grab my vaginal wall and I dissect that paravaginal space by tenting up on the anterior vaginal wall in a vertical plane, very similar to how we did your mesh graft repair. So you're probably very familiar with that, entering that paravaginal space. So I make my incision once I've done my hydrodissection into the vertical incision through the level of the vaginal mucosa and then the fascia, getting into that subfascial plane. And then I grab with Alice clamps and I dissect that paravaginal space with both my Metzenbaum scissors. And then I sweep that adventitia from a lateral to medial positions, kind of staying away and making sure where my bladder is. And I go sweep into a lateral medial just to get into that stay in that paravaginal space, staying away from the bladder and get to my ischial spines and along the sacrospinous ligament. So I'm again going to the ischial spine, sweeping that adventitia away and sweeping all that extra tissue to get to my sacrospinous ligament. Once I get to that sacrospinous ligament and I have that tissue swept off, I place my anchoring device in that lower medial aspect, kind of 1 or 2 finger breaths medial from the sacrospinous ligament. And I want to go into that middle lower third of the sacrospinous ligament. And we use an anchoring device. You can use a Capio needle driver, the Anchor Shore, which is very popular anchor device, and it has a O-proline suture that is placed on that. So I place one on either side. So I'm doing that same dissection on either side of my paravaginal space. I'm placing my anchor with a proline suture so that I have an anchor on either side. Then I go to the back table, I fashion my graft, I measure, I have a template, which now I measure my graft. If I'll go back to that picture, I measure this 4 centimeters in width. And so I also want to look at the patient too. They may have a larger surface area that I have to cover, but standard wise, I measure 4 centimeters width so that I have enough length so that I can actually draw out my arms. And these arms are anywhere from 6 to 8 centimeters once I've measured them. So then once I've done that, I then can place the proline sutures, and I usually use empty Mayo needle with my needle driver through the graft of the arm on either side. So I'm taking each proline through and through the graft to probably 1 centimeter from the edge so that you have a good hold of that arm. And then we tie down on either side the graft and I eliminate any suture bridging. So we're tying down the proline, tying down that graft arm directly onto the sacrospinous ligament. Then I use a 2O proline, PDS, I'm sorry, to suture distally. So we're suturing the ends of the graft at the level of your urethrovesical junction to that arcus tendineus fascia pelvis, that white line laterally. And I've sutured that 2O PDS. And you can use, if you wanted to use a proline, you can, but I'm tacking that suture to that graft. And I also use that 2OPDS to suture in the graft, both apically and distally in the midline, but creating a tension-free graft repair. Once all that is in place, then we're closing the vaginal mucosa, very similar to when we would do our mesh repairs, full thickness with fascia and vaginal mucosa with a 2-0 vicryl, and I do an alternating locking repair to close the vaginal mucosa. So you can see again, I'm tacking the 2-0 PDS here on either side to my arcus tendineus fascia pelvis between the bladder and the vaginal wall. And then my 2-0 PDS I tack here midline. And if the patient has a cervix, it can really get into that cervical stroma. And then the 2-0 PDS, sometimes I'll tack right here at the level of the urethrovesical junction. Then once that is all in place, again, we close our vaginal mucosa. I want to thank you for your attention today. And if you have any questions or comments, please feel free to email me. My email is here. And or if you want to email Mackenzie DiPietro at Allosource for any questions you may have specifically on the material or the graft or how it's, how it is prepared, please. And I want to thank you again. You have a wonderful rest of the Congress and appreciate your time.